Hello, everyone, and welcome to our next session for the Rosalind Franklin Society end-of-year virtual meeting, The Future is Biotech. My name is Dr. Phelan, and I'm Senior Editor of Gen Biotechnology, the new peer-reviewed journal launching in early 2022, publishing outstanding original research and perspectives in the biotech field. It's my honor to be joined today by Dr. Hannah El-Samad, Professor and Vice Chair in the Department of Biochemistry and Biophysics at the University of California, San Francisco, and Editor-in-Chief of Gen Biotechnology. Dr. El-Samad is the recipient of numerous prestigious awards, some of which include being named a former Packard Fellow, a Paul G. Allen Distinguished Investigator, and a Senior Investigator of the Chan Zuckerberg BioHub. In this session, Dr. El-Samad will share with us who she is as a scientist and describe the pressing need to uplift, amplify, and promote diverse voices in the field of biotech. So Hannah, it's so great to be with you here today, and we look forward to your presentation.
Okay, thank you. Thank you, Faye, and thank you all very much. It's such an honor and pleasure to be here with you today. Thank you all for joining. So for the next few minutes or so, I would like to talk about a set of topics, scientific and otherwise, where my passions really lie. I am a scientist and an engineer, and for a couple of decades now, what has driven my science at a very deep level is this desire, this urge to understand how life and the living exhibit this truly bewildering precision and resilience. As we carry out our lives, our existence, systems in our body, from our cells to our organs, stay in equilibrium. They keep their measure, they keep precise measure, despite all the turbulence around us. So this is called homeostasis. And this remarkable homeostasis is one of the deepest underpinnings of life and why it exists on Earth. And it finds its roots in something called feedback control, which is a technical notion, but also a very intuitive notion that we experience in our everyday lives. It's this ability that goes from our cells to our organs, to our whole body, to our brain, to always be looking out for the world, to sense what's happening around us, to compute with that information, and then to use that to react and adjust to the world. So as I said, every cell in our body does that all the time, 24/7, for the totality of our life. And it's all coordinated to generate life. And that is the question that has fascinated me for a very, very long time. Needless to say, a day without this ability of feedback control is a bad day. It's a bad day for a cell that cannot count its chromosome, that starts proliferating out of control, that cannot fix the damage that it incurs. But it's also, a bad day all the way to ecosystems or even our climate. And it's all of its current woes because these feedback control loops are starting to break. So my lab has spent the last couple of decades trying to understand, at the cellular level mostly, these feedback control systems. How do they work? How do they fail? How does their failure result in disease? Can we fix that? How do we fix that? And most recently, can we actually build them from the ground up to try to understand the most fundamental principles of their operation? To do so, we have been operating at the intersection, at the nexus of two very exciting fields, the fields of systems biology, the study of networks, the networks of life, basically, of cells that make cell function, and synthetic biology or cell engineering, which of course is is part of the broad area of biotechnology. We asked in multiple contexts and in multiple systems and cells and organisms, whether we can understand the complex networks, feedback networks in cells, so that we can rationally engineer them and fix them when they fail. But we also try to engineer these feedback control systems to change their properties. And through this inquiry that goes back and forth between these different fields, we have been able to make many discoveries and inventions. I'm going to just go through a very small number of them. So in the area of can we understand these feedback networks, we actually discovered fascinating new functions of these biological feedback systems. For example, how cells use them in processes of differentiation to shift the threshold at which these differentiation processes happen so that the organism is tuned to its environment. If the environment is going well, then you differentiate. If not, you don't. So as a way to sensitize a differentiating organism to its environment. We also discovered in this work that cells use them to play the lottery so that they can hedge their bets, to make their operation more stochastic, more uncertain, so that they can hedge their bets against environments that are uncertain. In the domain of can we fix feedback control systems, we actually did, and this is one of my favorite projects ever. It's been called the cyborg cell because it interfaces a cell with actually a computer. What we did is we took cells and we broke some of their feedback systems. So they lost the ability to sense and correct to their environment in certain of that in some of their processes. And then we interface them with a computer that can read out the broken processes. And then the computer decides how to correct for the deficiency and on what time scale, and then feeds this information to the cell through a process called optogenetics, a biotechnological process called optogenetics. So in this way, we were able to actually figure out the requirements of the missing feedback control. And to put it in a much simpler way, let's say that a feedback process in a cell breaks and you get a disease. What we asked was when and where and how much do we need to get the therapeutic intervention to fix the problem precisely? And so that was so much fun, and we're still doing a lot of that work. And more recently, we went all synthetic. So we call in building, these systems from inside the cell, with biological components. And we collaborated with the protein design pioneer David Baker to produce a number of de novo design proteins. And what that means is that these are proteins that don't exist in nature. They're designed computationally on a computer and then plugged into cells. And the amazing part of this is that they actually work, even though the cell has never seen them. Actually, these proteins have never existed before. One of these proteins is called Dagron locker. It's a switch that switches the cellular operation on and off. And we also used it to build feedback control systems that allowed us to change behaviors in cells and so on. So stay tuned for much more of this work. And the reason I'm mentioning it here is that in the context of biotechnology is that we anticipate that these capabilities will be so enabling to many branches of biotechnology to make the operation of engineered cells, whether it's for environmental purposes or therapeutic medical purposes, much more robust and reliable through this abilities, through giving them this ability to sense, compute, and correct. So with that, what I really would like you to take out of this talk, and the science part of this talk at least, is that cells are incredibly programmable. So this is exciting. This realization that not only can we change, genes, a single gene inside the cell, but we can upload whole programs, whole circuits inside the cell, makes for a very, very bright future. we can imagine, at least I can imagine, a near future in which we can actually fix cells inside in this way to repair their feedback control systems or their homeostatic systems inside a living body. So, as I said, I believe this future is possible, and it's hopefully around the corner. But for this to happen, we actually need all our talent pool. We need a lot of talent. We need to capitalize on the brain power of everybody in this country and around the world. So are we using all of our talent? So that's the next question I want to address. Are we capitalizing on all our talent? Of course not. I don't need to elaborate on this point because the facts and the numbers are known. Here's just one example, or a set of examples. It should, as you look at the slide, it should alarm you, it should alarm all of us that the talent of women is still undercapitalized. It's still under-recognized. We discard their scientific work. We don't cite them. We shun their recognition. We don't want them, give them opportunities for funding. We don't give them awards when they excel. It really pains me. It really kind of cut really deep when I made this slide. This is not opinion. These are numbers. And it gets even worse. In some areas, we're really going backwards. We used to graduate 4.9% of our engineering graduation, or people who graduate from engineering used to be Black and African American. In 1997, it dropped to 3.9% in 2016. Black, Hispanic, and Native American combined make up 5.1% of the workforce, the senior intramural workforce of the NIH. So this is a topic that really keeps me up at night. We have to do better, and we can do better. And one way to do better is to call on people who are in charge and call them to task. So I am a member of many groups that think about these issues. And one of my close collaborators in this domain is Dr. Lola Eniola Adefeso. And together we wrote a letter to Director Lander, the director of the Office of Science and Technology, to the president. And we asked him in this letter to take concrete steps towards fixing these issues, which are not theoretical, and not in need of more research and polling, but in need of concrete and urgent action. So we are still waiting to see what concrete steps happen on the ground. And these steps need to be bold. Make no mistakes, they need to be bold. They need to be urgent. So I'm going to put here for you just one of these ideas that we've been floating. And that's what we're calling the American Innovation Pledge. And what this means is that public agencies and private organizations, along with industry, come together to negotiate and sign a pledge that will move the needle on representation, support, and valuation of women and minoritized populations in STEM fields. So it would be like a big roadmap where everybody has their part to do, and they pledge to do it. They can pledge monetary amounts, human resources, strategic resources, that they all come together to accomplish so that we can bring forward every talent we have in this country to the table and to the fold of innovation. So the second idea I want you to remember is that bringing everyone to a level playing field is, of course, it's a moral imperative, and this is enough. Being a moral imperative is enough to make it actionable on its own. But lucky for us, it's also an economic an innovation imperative. Because study after study, science shows that diverse teams are more innovative. So biology, hopefully, or biotechnology, as I hopefully argue to you, in all of its forms, offers a bright future. I also argue to you that to achieve its potential, we need to give everyone in our society the opportunity to excel and to amplify their scientific voices. So let's make this happen. Because it's a moral imperative and because it's an economic and innovation imperative together. What's stronger reasons do we need to make it happen? So this drive to make this progress happen is part of why I, as Faye mentioned, this is why I decided in large part to be at the helm of the new Gen Biotechnology Journal. So to take my shot with an excellent editorial team, Faye is our senior editor. It cannot get better than that. And an excellent institution to make this a reality. So at Gen Biotech, we are interested in covering all exciting discoveries and progress of biotechnology, publishing the most scientifically rigorous, innovative, and groundbreaking progress in the field. Biotechnology is a necessity for our future to tackle our environmental and health challenges. There is no question about that. So what do we need for innovation? We need diversity. And this is what we're going to do in this journal also. We're going to bring diverse opinions, diverse voices, diverse ideas. We are starting, of course, with an incredibly deep, rigorous, expert, and of course, incredibly diverse editorial board. I'll be scrolling through some of, you'll see a lot of faces and names that you might recognize, amazing people. I don't know who to highlight, but this is Dr. Lola Eniola Adefesa, who is my thought partner with respect to the DEI, but she's also, first and foremost, a pioneer in novel materials for drug discovery and delivery and imaging. This is Dr. Tejal Desai, who was named today the new Dean of Engineering of Brown University. Who do I mention on the slide? It's an abundance of choice. Oh, Dr. Princess Imakude, who is the incoming chair of the bioengineering department at the University of Washington, Seattle, and a pioneer in biotechnology with an emphasis on women's health. And of course, Dr. Ana Maria Porras, who is a new faculty at the University of Florida, who is just building an incredible research program in engineering of in vitro models for the study of human microbiome interactions, but she's also an incredible science communicator. You should follow her on social media and see her impact and the education she does to the public on many bioengineering and biotechnology issues. And of course, shout out to our new associate editor, Dr. Alison Sgro, who is a faculty member at Boston University and a rising star in multicellular synthetic biology and cell engineering. So, of course, I highlighted only a few. They're all of our editorial board. They're all superstars. We're only getting started also. The editorial board would grow and reflect the depth and diversity of talent, opinions, and science in the field of biotechnology. We're preparing an incredible first issue of the journal, so stay tuned for that. Send us your papers, your opinions, your ideas. I really do hope that this journal will be the meeting platform for this whole, exciting and hopefully diverse field of biotechnology. And with that, I'll stop sharing my slide and see if Faye has any questions for me.
Thank you, Hannah, for that fantastic presentation. And we are now going to transition to our Q&A session. And I think my first question for you is that you mentioned this fantastic editorial that you published in Science, talking to Dr. Eric Lander about the current state of systemic discrimination in STEM. Did you get a response from Eric Lander after publishing that piece?
We did indeed, actually. So the moment the piece appeared, he tweeted quite a bit about it. He had a very thoughtful, actually, Twitter thread calling on everybody to read the editorial, the letter, and to think very deeply about what we can do to fix the problems that we highlight. We also had a number of meetings with him and his staff where we explored a number of ideas. So we are hoping that some of these ideas will soon materialize into actionable proposals and implementations. Our editorial was, our letter was titled Director Lander, the time is now. And now I'm going to upgrade it to the time is still now, but we need action. We might be running out of time. So we still have a window of opportunity, but we need to really explore it productively.
Yeah, that's great. And I think that leads into another question that I have here about action items and how, for example, you've done a phenomenal job raising awareness about these issues of diversity, equity, inclusion, STEM. How can other individuals in the biotech field, either academia or industry, do their part to also advance this mission?
It is actually quite simple. We all can do something within our circles and within our capacity. Stand up for innovation through diversity in whatever capacity you can, whether it's calling people up on their biases, making sure you have a fair and bias-free process in your hiring and your reviewing paper and reviewing proposals on panels. If you're in a biotech company, I say you're lagging behind. Look at the amazing talent you are missing out on, you're leaving behind. Whether when you appoint homogeneous boards for your biotech or in your hiring, if you're not intentional about the diversity in your hiring. If for those who are in a position of power and privilege, should use it. Use your privilege. Use your voice, use your resources. Many are doing so, actually. I invite you, for example, to see, and this is an example I know of, to see the program that the Packard Foundation recently started, the investment they made for increasing representation in STEM. So let's all get together and implement this American Innovation Pledge.
What advice do you have for other women and people of color facing underrepresentation in their fields?
I have a lot to say on this. I'm going to try to be brief. Something that I've lived by for a while and whose origin I cannot attribute anymore because a lot of wonderful women in my circles keep, saying the same thing, which is this quote that goes like this: if they won't give you a seat at the table, bring your own table, which is so powerful. This is to say that sometimes it's better to build a new system, to build a new journal, to build a new society, scientific society, rather than try to fix slowly and painfully irredeemable systems, right? So I consider actually the Rosalind Franklin Society an example of that. So gather your people and build a beautiful, shiny, innovative, diverse table. So the other advice I would give is, and this is really important, and I hope a lot of people hear that and believe it. You are who you are where you are, and you got the chances you got because you are talented, because you deserved it, and you worked so hard for it. Believe it, and do not let anybody tell you otherwise. This is really important for me to say. And finally, let's lift each other up. Let's never forget to lift each other up. My success, undeniably, 100%, couldn't have been possible without the so many women who lifted me up. And any success I have is insignificant if I don't lift others up with me along the way. So let's be kind to each other and let's lift each other up.
I love this idea of making your own table. And
I think isn't that cool? It's just so simple and so profound. So, so enabling.
Yeah, absolutely. If that space isn't there, you make it yourself and put your voice out there. And I think this also aligns well with you taking on the editor-in-chief role at Gen Biotechnology. So if you wanted to say a little bit more about what role Gen Biotechnology will play in advancing diversity, equity, inclusion in the biotech field, and what is your overall vision for this new table or this journal?
Yeah, so I think I already mentioned some of that in my closing slide. I really would like the journal to be the meeting place of the field. I would like it to cover all aspects, starting from a scientific perspective. I really would like to cover all aspects of biotechnology. The full ecosystem, its science, its sociology, its nascent talent, its challenges. I'm really hoping it will be a place, again, where a beautiful, diverse community gathers. So I'm hoping the content will reflect that, from cutting edge science to forward-looking perspectives to voices telling the field what is holding us back, whether it's scientifically or pushing us, propelling us forward, right? Whether it's scientifically or sociologically. Of course, we would like to feature diverse scientific and social voices who speak to different technological needs for our community and the world. I would really love it to be the voice of the young, those who will propel us forward. We should be the beacon through this journal, the beacon of the biotechnology field.
I love that. And with that, we are out of time for this session. Thank you all for joining us.
Thank you, Faye. Thanks, everybody.
Thank you, everybody, for joining this fantastic presentation with Dr. Hannah El-Samad, professor at UCSF and editor-in-chief of Gen Biotechnology. This has been the Future of Biotech for the Rosalind Franklin Society end of year virtual meeting, and we hope you will tune in for the rest of our program.